we talk about computational prediction of food functionalities and analysis of the mode of actions using chemical and medical data. Foods contain many constituent compounds that regulate biological functions. Thus, food would be potent for preventive health strategies and recovery during early disease stages. However, there are so many different foods, constituent compounds, and diseases that it is nearly impossible to determine the functionality of all foods through experimentation. In this study, we predicted the potential functionality of diverse foods for a wide range of diseases. Furthermore, we estimated the mechanism underlying the functionality of foods. Here, we will explain the methods. First, we inferred the interaction between constituent compounds and proteins on disease-related pathways. Specifically, we represented the chemical structure of the compound using chemical function and substructures descriptors. KCFS is suitable for representing the complex structures of natural products. Next, we inferred the interactions between 56,040 compounds and 4,873 proteins using sparse logistic regression. We then estimated the inferential relationship between 876 foods and target proteins. We considered that food function in diseases by regulating many proteins on disease-related pathways. Therefore, we mapped the target proteins of foods on 83 disease-related pathways and predicted the functional relevance between foods and diseases. The strength of functional relevance was indicated using the relevance score. We evaluated the accuracy of the prediction of functional relevance between foods and diseases and used AUC scores as the evaluation metric. The mean AUC score for foods was 0.76, while the mean AUC score for diseases was 0.64. Here, we will explain the results. We applied the proposed method to 876 foods and 83 diseases. We evaluated the 20,778 predicted functional relevance. The functional relevance was visualized in a cluster map based on the relevance score. In the 2D block, there was functional relevance between foods such as tea, citrus fruits, and animal products, and neurological disease and endocrine diseases. There have been many studies on the relationship between citrus fruits and tea and neurological diseases. The consistency of our results with previous studies emphasizes the validity of the proposed method. Furthermore, we inferred the mechanism of action of food functionality. We visualized the relationship between food, constituent compounds, target proteins, and diseases using a network. The figure shows the mechanism of action of grapefruits for Parkinson's disease, insulin resistance, Alzheimer's disease, and dyslipidemia. The dark orange lines indicate known interactions. Many known interactions between the constituent compounds and the target proteins were included in the figure. This suggests that the proposed method is useful for inferring the mechanism of action of foods. Generally, people consume multiple foods at the same time. Therefore, we finally investigated the combination effect of foods. Food combinations were explored based on the filling rate of the disease-related protein targeted. The estimated food combinations for 4 diseases are shown in the table. In summary, the proposed method enables us to detect functional relevance between food constituent compounds and diseases, and suggests the mechanism of action and beneficial food combinations.